Thank you for introduction. I'm Masayuki Karasuyama. I'd like to first thank to the organizers to present this great challenge and give me an opportunity to talk here. The title is the Learning Kernel-based Feature Representation for Gene Essentiality Prediction. So here's our team. The members are I and Professor Mamitsuka. We are from the Bioknowledge Engineering Research Laboratory in Biomathematics Center, Kyoto University. We are studying the data mining and machine learning approaches for biological data analysis. And personally, my major is the machine learning methodological detail. And this talk, mainly I will describe about the methodology I employed in the challenge. So first, I would like to quickly review the problem setting. The task is to predict the essentiality using the gene expression and the copy number and the mutation. So this task is, as a machine learning problem, this is a multivariate regression or multitask learning. And the final accuracy is evaluated by using additional 44 cell lines. So the goal for participant is to achieve the higher accuracy for these additional 44 cell lines. So for this problem setting, we focused on the following two difficulties. The first is the output variables, that is essentiality, contains a huge number of variables which are highly dependent to each other. So this dependency should be incorporated into a model. The second difficulty is the number of sample, that is, number of cell line, is quite small compared to the input dimensionality. So for these problems, our approach is that we employ a kernel-based prediction model, so-called kernel-rich regression, and try to incorporate the dependency, output variable dependency into the kernel or feature using the kernel canonical correlation analysis and the kernel target alignments, as we will see later. And we call these approaches learning feature representation in this talk. And for the second problem, to alleviate the small sample problem, we learn only one shared feature representation using all output variables. So first, let me confirm some notation. Xi represents the input feature vector, which corresponds to the column of this matrix. And yi represents the essentiality vector for about 15,000 genes. This also corresponds to the column of this matrix. And n represents the number of cell line, which is equal to 105 in this case. So as a base model, we employed a kernel-rich regression, which is a quite standard prediction model. And actually, this is almost equivalent to the Gaussian process regression, which is often appears yesterday as well. And the prediction model can be written by the this equation for the case gene essentiality. This equation is a linear combination of the kernel function k, which combined by the parameters alpha. And these parameters can be learned by the standard regularized least squares using the quite small linear system. And also non-linearity can be incorporated depending on the choice of the kernel function. For example, famous Gaussian kernel. So this approach worked reasonably well in some sense, but kernel regression, KRR in short, does not consider the dependency among different output dimensions, different genes in this case. This approach just solves each gene independently. So however, the essentiality of different genes are highly dependent to each other. So actually, we verified this fact by learning a model predicting gene essentiality from essentiality of other genes. And I confirmed this achieved much higher accuracy. Roughly speaking, it achieved the 0.4 Spearman correlation, which is much higher than the final top-ranked score. So we tried to incorporate this dependency into a kernel or feature using the kernel-based feature representation learning, in particular using the kernel canonical correlation analysis and the kernel target alignment. So this is the illustration of our system overview. The input data first substituted into a kernel canonical correlation analysis and the kernel target alignment. We call this process learning feature representation, and they produce two kernel matrices, as we will see later. And using these four kernels, we run the kernel-rich regression, respectively. And finally, take average as a final prediction. So I mainly describe about fast learning feature representation process in this talk. So the first is about kernel canonical correlation analysis, KCCA in short, which is a quite standard feature extraction or dimensionality reduction method, which projects X and Y into a common subspace in which projected variables are maximally correlated each other. In this illustration, X and Y projected into a U and V, which is lower dimensional compared to the original X and Y. And as a result of this projection, dependency among Y is embedded into lower dimensional variable V, to which U is maximally correlated. Thus, in other words, we can expect U can explain the dependency among Y. So we run kernel distribution model using the projected variable U as a new input data, as like this illustration. So by which the dependency among Y can be incorporated into a model. And this kernel canonical correlation analysis process can be kernelized like usual kernel method, and this can be solved easily, though I don't go in detail the optimization. So the next approach I employed is the kernel target alignment. And this is the method to run a kernel matrix. Actually, this method runs a kernel matrix so that it can approximate so-called ideal kernel. In this case, we define the ideal kernel using all dimensions of Y. This is the illustration in the case of the linear kernel using all dimensions of Y. So predicting essentiality using this KY means that predicting essentiality using essentiality of all genes. So that is predicting essentiality using dependency among different genes. But the problem is KY is not available for test point, though the KY contains strong prediction power, but we cannot, KY is not available for test point, so we consider approximating this using the input data. So we approximate KY using this matrix, psi transpose M psi, where psi is the input data projected into kernel feature space. That is the psi transpose, psi is kernel matrix. So, and we maximize the correlation between the ideal kernel and this matrix regarding the M as a parameter. And this is again that can be kernelized and can be computed quite efficiently. So again, here I don't go in detail the optimization. So kernel CCA and kernel target alignment are the different methods, but we can interpret them from the common one picture in this illustration. They are similar in a sense that they both run the kernel or feature using the all variables in the output. So like this illustration, so we can, dependency among a high-dimensional output can be incorporated by running the kernel or feature, unlike just running kernel regression independently. So, and also, by running only one shared kernel, random kernel is used for all output dimension. By sharing the common kernel or feature, we also expect to avoid overfitting. So I would like to briefly show some experimental result. For input data, we use the Gaussian kernel to incorporate nonlinearity. And also, we have an option for the output kernel. In this case, we use the linear and the Gaussian kernel. So by using the linear kernel, Gaussian kernel for kernel canonical cohesion analysis and the kernel target alignment, as a result, we have the 4 kernels and run the kernel regression respectively. So here's the some settings, implementation details. For Gaussian kernel with parameter, we employed well-known median heuristics. And for regularization parameter in the kernel regression, we determine it based on preliminary experiment using the cross-validation. And we use expression and the copy number, and we do not use mutation data. And expression has the strongest prediction power as far as I observed. And also adding the copy number could improve accuracy somewhat. So in general, simpler, that is the low variance approaches worked better for this small sample problem. So here's the performance evaluation using the final test set. And the bottom line, average, is actually our final submission. And the first line is the naive application of kernel regression. And the next 4 methods are the model applied each feature representation method. And except for the kernel CCA with linear kernel, we observed that these methods could improve the accuracy of the base model. Actually, kernel CCA with linear kernel also improved the accuracy in our preliminary experiment, but in this case, it couldn't. This might be some because of the perturbation of the smallness of the samples. So actually, we took the average to avoid such perturbation, and in fact, we can see the average output is better than the all other individual predictions. So let me conclude my talk. We developed a gene essentiality prediction algorithm based on the feature representation learning. And incorporating the dependency among different genes, we improved the accuracy compared to the naive baseline model. And also, our approach is computationally quite efficient because we don't need to access to high-dimensional input data after once we calculate the kernel matrix. So that's all. Thank you for your attention.
Thank you. That was very interesting and elegant work. Thank you. It was, I think the idea of using the correlations among very high-dimensional response variables is something we've seen in a few challenges with the winning team. So it's nice to see that kind of consistency, and I thought this was a really nice approach. So the second sub-challenge, this one was focused